Hello, we're glad you've joined us for this live webinar, not your typical flow cytometry panel design webinar, a toolkit for selecting antibodies with the complete panel in mind. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration. It's sponsored by Beckman Coulter Life Sciences. To learn more about our sponsor, please visit www.beckman.com. Let's get started. You can post questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing a presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Ask a Question box located on the far left of your screen. I now present today's speakers, Maria Gentile and Michael Kapinsky, both Senior Marketing Managers at Beckman Coulter Life Sciences. To learn more about our speakers, please visit the Speaker tab at the top of the presentation window. Maria and Michael, you may now begin your presentations.
Thank you, Michelle, for that kind introduction. Michael and I are really happy to be here to talk together today, and we'd like to also thank the audience for being here. We've been working on this collaboration for a while, and we're really enjoying the idea that we can bring concepts together for both developing panels and the instrumentation's contribution to that. So, here we are today, and we'd like to really focus on, why it isn't always advantageous to use the brightest dyes for every marker in a panel. How do you then incorporate the instrument characteristics into your panel design? And then, also demonstrating how you can apply good panel design and in a way that helps both really new to flow cytometry users as well as advanced users, and how it makes it easier for you then to communicate how you've made decisions on how you put your panel together, and then also learning some things about technology and how the technology really can influence the decisions that you make when developing a multicolor panel. And so with that, Michael, I think I'm going to hand it over to you.
Thank you, Maria. Yeah, as you already said, today we really want to convey a very comprehensive view on what you would consider when building a panel. So really looking at the whole system that we want to optimize, not only picking several features and work on those, but really considering the complete system. And of course, the fluorochromes are important here. So let's start with the fluorochromes and look at their brightness, but also considering what else is a characteristic of a fluorochrome, for instance, the spillover. So what we see here in this scheme is a relatively limited selection of dyes that also will be subject to our examples during the next slides. As we go from left to right here in this picture, we will see that the dyes that have been ranked here have an increasing relative brightness. So from the very left, for instance, where we see on the violet laser chrome orange, we have a dye that you typically would use only for antigens that are very densely expressed. And as we go to the very extreme right, we'll find PE, which is probably the brightest antigen here in this scheme, that could be used with any antigen. It's bright enough also to be used with antigens that have a very low expression density. Of note, this ranking here is really just a qualitative ranking. So there will be differences in how these dyes are relatively bright to each other if you consider different clones or different conjugation chemistry. Sometimes the formulation of the antibody may also play a role. So we just have seen that there is a broad range of brightness between dyes, from really dim dyes to very bright dyes. And the same, of course, holds true for antigen densities, if this is not a much more extensive range even. I put some examples here together. Let's start maybe with the human T lymphocyte CD4 expression in the lowest row in this overlay plot. Very densely expressed, around 150,000 copies per cell. And you see that the characteristic of this expression is bimodal. So we'll have a very bright, a very densely expressed CD4 on a positive population, and we'll have a negative population. If we now go one row above, we see a modulated expression profile. So here we will have events that have a very dense expression, maybe even beyond what CD4 is. But also, we will see events that have an intermediate expression density, as well as events that have a very low expression density. So this is probably the most challenging antigen to implement here, and we'll see during the next slides. So I hope I can show that, why that is. Let's go one line further, where we see a weak expression density, a negative population, and a positive population. However, we only have low expression densities here in this positive population. And then even one further, what we labeled here, you need to believe, is that you not only have very low expression densities on a positive population, but you also have a very low frequency of these events. So here you really need to make sure that you recover all the events that are in this low positive range. Example would be, for instance, stem cell-like T cells memory type that have a very dim CD95 expression on a very small portion of the T cells only. So let's go back to the CD4 example. What I've shown here on the left is a CD4 strain, and you may also remember the dichrome orange to be one of the dimmest in our range of dyes. The result that we get is, however, just bright enough. So we see all the CD4 positive cells, and we also see very clearly separated the CD4 negative population. On the right, we get exactly the same result when using a very bright dye. So again, we have a separation between CD4 negative and CD4 positive. However, the separation is much more pronounced than on the left. But still, the result that we get is comparable between left and right. But on the right, we see also that the positive population starts to get broadening as compared to the negative population. So in this channel on the y-axis, the 620 nanometer channel, we will see an increase of measurement error called spreading on this CD4 PE positive population, which limits the sensitivity for an antigen measured in this channel that we can achieve on the PE positive population. So brighter is not always better. We really have to understand that brightness is helpful. Here in this case, we see that brighter might be in a disadvantage, and it may be advantageous to have less brightness on the CD4 positive population. So to summarize, be just as bright as needed, but be also as dim as possible. That could be a takeaway here from this picture. So this brings us to the Goldilocks principle of dye brightness. We have two extremes here. We have a very dim expression, very weak expression, and a very strong expression in the top of the slide. And we now want to choose a dye that allows us to have enough sensitivity, so not be too dim for a weakly expressed antigen, but at the same time, once we consider a strongly expressed antigen, we don't want to have a dye that is too bright, that causes too much spillover, that might impair the sensitivity that we can achieve on other detectors. So we really want to balance that. We don't want to be too dim, have enough sensitivity, but also not being too bright, so that we avoid spillover into other channels. And again, that is basically the same message. Be just as bright as needed, but be also as dim as possible, just find the right intensity to really meet the needs regarding your antigen. Just a real-life example here. Again, we see a CD4 stain on the x-axis. It's well separated, so there is not a lot of distance, but we can clearly understand what the CD4 positive population is. And without any spreading into the y-axis, we can see the expression of a very dimly or very weakly expressed antigen. So here we have to make sure that we use a very bright dye, the brightest dye that we have.
So that's a nice graphic representing the first principle, which is understanding what expression type you're dealing with and also balancing that with the right combination of Flurochrome.
Yeah, and let's take this now a little bit further. So we have matched the antigen density with the dye brightness, and we have done this considering the brightness of the dye, but also the spillover that it conveys to other detectors. And here's just an example of what that can mean in real life. So we have two 10-color panels here in this picture. In each of them, the same antibody clones were used. However, from between left and right, we have swapped the fluorochrome assignments, the fluorescent labels of the antibodies, except for CDX PC5.5. So this antibody that obviously detects a very weakly expressed antigen is the same in the panel on the left and on the right. However, the results that we obtain using the same antibody clone here, even on the same sample, of course, are very different. So on the right plot, we can only detect about a quarter of the positive events that we can detect on the left. And the reason for that is that the other fluorescent labels that are on the same cells of interest here impact the resolution that we can obtain on the PC5.5 channel on the right, while on the left we have done a very smart design avoiding these spillover effects into the PC5.5 channel, so we can have a very sensitive detection. So what we can take away from this, comparing this to staining, is that the sensitivity of detection for any labeled antigen is really a function of all the fluorescent labels that we find on the cell surface. So the sensitivity for each antigen will be determined not only by the fluorescent label that are used for this antigen, but also by the spectral surrounding, the spectral neighborhood, all the other labels that are also present on the same cell surface. And from a biological point of view, this just means the antigen expression pattern will be very important regarding the sensitivity that we can achieve for a given antigen. So let's try to put this a little bit together. We have seen Flurochrome characteristics, brightness, spillover. We now want to take the biology also on board to put this together. And furthermore, of course, we will look into the way that a certain instrument, that a certain detector will detect, how a certain detection system will detect fluorescent signal and how it will process that, because this also will have an influence on how we think panel design, and it will especially be important for the stringency that we have to apply when designing panels. So when we want to talk about the biology in a certain antibody panel that we want to apply, it is very efficient to use antigen trees like we see here on the right. In an antigen tree, we can implement all the relationships between the antigens, and also by color coding this here, we can show the expression profiles that are associated with these antigens, as we see on the right. I remember we have used this depiction a couple of slides earlier already. So for instance, a green antigen in the antigen tree is a densely expressed by modal antigen, where, for instance, a purple one is a weakly expressed antigen. So to understand this tree and what information is inside this tree, maybe we start with looking at the parent-child relationship. So a child antigen would also always be co-expressed together with its corresponding parent antigen, whereas a parent antigen may well be expressed without this child antigen, could be expressed with other child antigens also, giving rise to various subpopulations. A common example for this situation is the expression of the CD3 antigen, a principal T-cell antigen, and then you might imagine a second antigen named CDX here that could be a modulated antigen exclusively expressed by T-cells. Furthermore, let's consider two dyes for our example here, like ECD, PE Texas Red, a dye with an intermediate brightness, and a very bright dye, PC 5.5. CD3, as an additional information here, causes spillover spreading into the detector where we would want to detect PC 5.5, while vice versa, this is not the case. And this is indicated here by the gray triangle that you see along the quadrant and along the y-axis. So following our idea of matching antigen density with fluorochrome brightness, we have chosen here on the left the ECD dye to detect CD3, so the intermediate brightness, and the very bright PC 5.5 we have chosen to use the co-expressed antigen, the co-expressed modulated antigen here in this case. And what we see in the gray triangle, that we would find dots in there that are gray, that are green, but also other dots that are red. So this indicates here that we have a certain range on the log scale where we would not be able to distinguish between green dots that make part of the negative population and just lie in this range of the scale because of the measurement error, the spillover spreading of ECD into PC 5.5. But also we see red dots in there that represent events that have a specifically positive expression of CDX. But we will not be able to distinguish between those. So what we end up with here is a loss of sensitivity for those low expressors. On the right, we have swapped the fluorochromes now, and now we have chosen the stronger dye for the lineage antigen and the co-expressed dye for the intermediate brightness antigen, with the intermediate brightness dye. However, what we also see that there is no spreading from the PC 5.5 dye into the ECD channel. And this allows us to have a very clearly resolved cutoff of negative versus dim positive. And so we will be able to detect all those CDX positive cells that have a very low antigen expression density, which gives us overall the better sensitivity as compared to the case on the left. So the takeaway message here is that spillover from parent antigen into a child antigen should be avoided. Even this may override other rules like the antigen density fluorochrome brightness rule. On the right, we have made sure that the spillover here only occurs from the child antigen into the parent antigen, and this gives us the possibility to have a much better resolution and a much better sensitivity than seen on the left. So what does this mean in real life? Let's look into a real example. On the left, we are again with the CD4 strain. We have considered fluors only, so the PE would cause a lot of spreading in the ECD channel, and we would maybe concern not to have PE and ECD labels that could occur on the same cell. However, on the right, we have taken the biology also into account. And using here a B-cell lineage marker, CD19, and a co-expressed marker, that is the immunoglobulin light chain lambda, that we would find exclusively on the B cells, arranging this now in a way that the parent antigen would sit in the panel that receives the spillover from PE. We see that there's no spreading problem occurring. We don't have PE positive events here that would not carry the B cell antigen, and this allows us to use this combination without any impact on our data quality. So why would we consider that? Well, of course, when we use a lot of fluorochromes in our panel, really use every channel maybe on our instrument, then we will use dyes that have spillover. So we have to make sure that we deal very economically with those pairs that do not have any spillover at all. And so we would want to identify biological expression patterns that would tolerate the use of fluorochromes that have a strong spillover. So implementing this into our antigen tree, we, for instance, could use these arrows here, a green arrow that indicates that spillover would be allowed, and a red arrow that you should avoid spillover when designing the panel. The arrows that are in this tree now would indicate the interaction between different hierarchies in the tree. So maybe have a look at the parent to understand how that works. Into the parent, or pointing into the parent antigen, we now have a green arrow. So this would tell us that the parent can well receive spillover without any impact on data quality. But at the same time, we have a red arrow that points out of the parent down to the next hierarchy. And this, according to our parent-child concept, would say, please avoid spillover from this parent into its child antigens.
That makes it really clear. I really like this graphical representation, and it really helps when understanding that the principle for what you can allow to spill over and what is to be avoided. And so when moving up the tree, spillover is allowed, and moving down the tree in levels of the hierarchy to avoid spillover.
I think that is a very crisp way of saying it. Just thinking of this going down and going up principle and how this is corresponding to the spillover spreading allowance or avoidance rules. Mm-hmm. So now let's think how we can think about spillover spreading effects that may occur within the same hierarchy, not only between hierarchies. And to do that, I think it's a good model to have a look at modulated antigens that have cells that have a very low, with cells that have a very low expression density of the antigen, and cells that have a very high expression density of the antigen alike. What we see here are the plot on the left and on the right are taken from the same data set. Actually, we are looking at exactly the same T cells here, and we look at the expression of CCR7 on the y-axis, CD197, and we have co-expressed checkpoint markers here, and all of these antigens have a modulated expression profile. On the left, we have made sure that the dyes that we are using to detect those antigens do not cause spillover spreading in each other's detectors. And the result is that independently here from, let's say, the CCR7 expression density, we can detect CD366 TIM3 with the same sensitivity. So regardless of a strong or a weak CCR7 expression, there's an independent sensitivity for TIM3 is independent from that. And in contrast to that, we see on the right what often is referred to as a spreading trumpet. So here, PC 5.5 causes a strong spillover into the PC7 channel where we want to detect the co-expressed LAG3 or CD223 antigen. And as the CCR7 expression increases, also the PC5.5 signal, of course, gets stronger. And this means that we have more spillover for a high PC5.5 signal into the PC7 channel. This translates to a larger spreading effect. And finally, in turn, this limits the sensitivity that we can achieve for LAG3 expression on those cells that have a high CCR7 expression. So that is definitely a situation that we want to avoid. We want to be on the left side where these two antigens can be detected with a high sensitivity independent of each other's expression level. Going back to our scheme, we now again will implement within a certain hierarchy of expression the green and red arrows. And we see that these arrows have been tilted a bit now to indicate that we are not talking between hierarchies here, but within the same hierarchy. Let's have a look at a certain element of the hierarchy to understand how this works now. Let's take child A. Child A has an arrow going out of the antigen. So we want to avoid spillover from child A to other members of the same hierarchy level, whereas the green arrow indicates that child A could tolerate spillover, and that is based on the nature of the expression profile that we see on the left. We have a very strong bimodal expression. So if we would have a spreading effect for the negative population, this will not keep us from detecting the positive population. In contrast, the purple color of child D indicates that we have a weak expression here. So the weak expression allows, as indicated by the green arrow, to exert spillover to other antigens of the same hierarchy, as the sheer amount of the spillover will be very little. So we can be confident that this will not impact the sensitivity. However, child D is very weakly expressed, so we must make sure that spillover into the channel where we want to detect child D must be avoided. So we will not put the detection of those low expressors at risk. So we see other elements also here in this antigen tree, where we have an exclusion and also a positive correlation. Let's have a look into what that means for our panel design. So we can start considering fluors only on the left, where we have two strongly expressed antigens, and we have made sure CD4 and CD8 in a T cell gate here. And we have made sure that we use fluorochromes that have very little spill over into each other. So this gives us a very nice picture, but we need to be really economic with those kind of fluorochrome pairs that really have no spillover, at the same time have a relative good brightness. We don't have an endless number of those pairings. So we may again also consider the biology in order to find identify the situations that would also allow us to use fluorochromes that have a higher spillover than seen on the left. And that is exactly what we've done on the right, using CD4 APC and CD8 APC Alexa 700. So these are two fluorochromes that whose emission characteristics have considerable spillover into each other's detector, as we also see by the spreading of the positive populations. However, we also see that the discrimination limits of interest are not in a range where they would be affected by this spillover. So again, we can detect CD4 and CD8 here with the same sensitivity on the left, despite we are using fluorochromes that have a very strong spreading effect on each other. So we can allow spillover between excluding antigens. It will not cause any problems for us. And this also holds true for a situation, expression situation, that basically is the opposite of what we just have seen before. Here we have two antigens that are not excluding each other, but two antigens that always occur together on the same cell. On the left, again, just considering the fluorochrome characteristics, we have chosen two fluorochromes that have no spillover or very low spillover spreading effects regarding each other detectors. And on the right, we have, in contrast to that, used fluorochromes that have considerable spreading effects. And because here we again have made use of the biology. So we know that those two antigens, just very similar like on the left, always occur together on the same cell surface. The only differences that we may see between cells and between certain samples is that expression densities may be modulated. So, and this is indicated here by this discrimination of interest dotted lines, which clearly indicates that we might just be interested here in a loss of a certain density of expression, but not in discriminating between negative and dim or weakly positive events. So like with the exclusion, in a situation where we have a positive correlation between antigens, we can also allow to use fluorochromes that have considerable spillover into each other's detectors. So adding this to our antigen tree now with the exclusion pairing and the positive correlation pairing, we can add an arrow here, a two-headed arrow that is all green, allowing or indicating that there is mutual spillover allowed for this kind of expression situation. So putting this all together, now we see the biology on the upper right here, where we see how the biology influences our rule of panel design regarding the spillover and the fluorochrome brightness. We have the ranking of the fluorochromes on the lower right. And now on the left, what comes on top of it is what we call a spreading matrix. Here in this spreading matrix, you would see that each detector is represented by a row, and each color that we want to use on these detectors is represented by a column. And as we now go down a column, we would see how a certain fluorochrome emission would cause spreading effects on these detectors that are represented by the row. And you see the legend below. So a pale blue would mean there's no spreading effect, and it would be scaled to dark red that we would see very strong spreading effects. So how does these three elements now can be used together? Let's have a look at some examples here. For instance, the parent antigen. So the parent antigen, as you see in the spreading matrix on the right, has been put into the chrome orange column. And in the chrome orange column, you see mainly blue fields, and some have a light shade of red. However, in those fields where we have a light shade of red, there are no antigens except for H, a modulated antigen, where we can expect a slight increase of spreading, but not dramatically. So we have been using a bright dye that has little impact on the other antigens for the parent. Let's take another example to show also the exclusion principle. Child A and child C are not expressed together, so we see them in the left on two places, Alexa 700 and Alexa 750, two columns that represent dyes that have a strong spill over to each other. You can see how A and C share sides of a dark red field. So A would cause a lot of spreading in the channel where C is detected. However, as A and C do not occur on the same cell surface, we can make use of this biological feature in order to use a pairing of dyes that may not be used in case where we would see co-expression of the dyes. So that is how this principle can work when we consider all features that we have touched upon. But I think it is important also to understand a little bit better what is behind this spreading matrix. And for this, I would like to hand over to you, Maria.
Thanks, Michael. I really enjoy, seeing all of these concepts come together in a graphical way. What great tools, right, for when you're designing a panel and you can actually understand the instrument and how the instrument can either help you or things that you may need to accommodate for. And so let's get then moving into that topic of what the instrument contributes and some considerations that you can make that might help you with your panel design. So the next part of the talk then, we're going to focus on the instrumentation and how instruments are responsible for managing light, and also where the software is important for not only controlling the instrument, but for providing tools that can also facilitate your workflow. And traditionally, I think it's okay to say that we've had to think about our instrumentation as what constraints does it put upon my experimental design. I think the newer technology, and specifically we're going to talk about the CytoFLEX, I think it's fair to say that you can ask a different question now. You can ask, what can my instrument do for me to help to make my life easier, help to make my panel design maybe a little bit simpler? Because as you could see from the concepts that Michael presented, there's a lot of principles to consider, and there's a lot of interplay between those principles. And so the CytoFLEX actually was designed specifically to tackle the challenges that the instrumentation might play in those considerations. And so CytoFLEX was designed to reduce noise and manage light, and we're going to look at how that works. So these are the two principles that I'd like to focus on. First of all, compensation doesn't cause data spreading, it only reveals it. And the measurement error is the root of the data spreading. So it's the root cause of data spreading. And so let's look at how instrument design can influence these two principles. Right. So first of all, we're going to focus on instrumentation, light management, and software. And how does the instrumentation contribute to reducing the measurement error, which in turn reduces data spreading? So the three components on the CytoFLEX that contribute to reducing measurement error, so the smart decisions made about the components and the design of the components that are on the instrumentation. One, the detectors that were chosen are avalanche photodiodes, which are known for having less noise, so having less measurement error. And part of that is because there's a small area upon which the signal is detected. Number two are the optics. So the optics are designed specifically to focus light better and reduce optical noise. And so it's a composite objective that accomplishes this. Thirdly, we need to think about the electronic noise. So there are controls put into place that help to reduce the noise from the digitization of the signals. And hence that is the way that signal is then translated into the way we visualize it within the software. And so bringing that into like a data visualization paradigm, we can look at two different types of instruments, right? So we have on the left an instrument that's PMT-based, and on the right an instrument that is APD-based, but specifically the CytoFLEX. And so using that example again of CD4 labeled with PE, we can see on the PMT-based detection system the spreading that is a result of the measurement error when the signal gets very bright, and you can see that spill over into adjacent channels. And then on the right side of the slide here, we're looking at that same antibody Flurochrome combination, but this time detected on a different instrument design. Okay, so there's an APD-based detection, and there's also other controls in place that help to reduce the spreading.
I think it's important to quickly touch upon what this, again, means really for the panel design. On the left, we have a situation with this considerable substantial spreading that limits the biological combinations that we can put here. So for instance, in this case, the detector that is affected by spreading would have to carry a parent antigen, or the detector or the dye that exerts the spreading to the other detector would have to be a dim expression. While on the right, where we don't have this restriction, there is no spreading. We basically are very, very free in what we want to put here, which co-expression markers or which other markers should go there. So it gives us a lot of flexibility, and there's very little restriction on the biological relationship of the antigens that can be detected in such a setting. I think that is really important to understand in order to link back how the instrument would really influence your ideas and thoughts about how to design the panel.
Exactly. That's such a good example. Thanks. So then, what else? We mentioned, so we talked about measurement error and controlling that and how that might help to influence the decisions that you make about your panel design. Now let's look at sensitivity. So instrumentation is also responsible for allowing detection of dimly expressed and some of those modulated expressed markers, as well as the ones that are so dim that maybe and so infrequent that you're really not sure that they're really even there. So what can an instrument do to help out with those situations? So the three components that I'm going to focus on next are the excitation optics, emission optics, and detectors again. The excitation optics, basically focusing on the flow cell. The flow cell was designed with the beam shaping optic integrated. And so we can think about every time the light has to move through or across a surface or be transmitted from one component to another, it's an opportunity for light to get lost and for us to lose efficiency in that light transmission. And so integrating the beam shaping optic into the flow cell reduces that opportunity, right? So it helps with the efficiency of the movement of light from your sample out of the flow cell onto the optics. And so moving on to the emission optics then, we can see the fiber delivering the light onto the wavelength division multiplexer, which is designed specifically and also to increase the efficiency of that light movement. And so as the light moves through a bandpass filter onto its detector, the rest of that light is reflected back onto a mirror. And so once again, there's no dichroics. Therefore, there's that minimizing the risk of light loss that would happen in the presence of that type of transmission of light. And we can also think of this as all of the light gets, this system is designed to collect all of the light, and then the software is smart in order to manage the interpretation of that light movement. And then once again, we've got highly efficient, high quantum efficiency photodiode detectors on board, which are known for their efficiency across a broad range of emission wavelengths, and also have a really good conversion of photons to digital signals. And the way we usually measure this, or a traditional way of measuring this, is by using a multi-intensity bead mixture that can display the resolution power across the different channels of an instrument. So what we're looking at here are 7 different laser options that are available on the CytoFLEX platform. So on the LX, you have an option for a total of 6 lasers, and you can choose between the UV or the near UV, as well as having an infrared laser on board. And so for the 6 of 7 lasers, we're demonstrating the resolving power using the rainbow calibration particles from Spirotech. And so this is a commonly used way of calibrating as well as aligning or optimizing the alignment of instrumentation. So we can see on most of the channels, we've got all 8 peaks resolved, and many of them are baseline resolved. So really good resolution power as displayed using multi-peak beads. And for the IR laser, what's shown here is a multi-intensity bead mixture of IR exciting and emitting particles from Spirotech as well. This is a really nice controlled way of looking at the resolving power of instrumentation. But what's really important is the biology, right? So how does the instrumentation contribute to the power you have to resolve these dimly expressed and modulated signals and the balancing act that you've got to think about when you're putting a complex panel together? I like to look at this data and say, things look pretty tight, right? I've got some really nicely, tightly distributed populations where it's not difficult to decide where to draw my gates. So from a data, from an overview data analysis perspective, I can look at this and say, I think, I can draw gates pretty easily and make my biological interpretations quite easily. But let's focus on a few of these plots, and I'm going to ask Michael to talk us through the biology here and the significance.
Yeah, thank you, Maria. So I'm really not the bleed-based person, so I really like that we see biologically samples here. On the left, we see the basophils very nicely separated out of the whole crowd of leukocytes. So, of course, there's two aspects here. On the one hand, the clones being used, the Flurochromes are really nice, but also the instrument allows here for a very crisp and sharp carving out of the basophils. We see the Tregs in the middle with the 127 and 25 staining, where I think you can see that these staining the CD25 high and CD127 population really is clustering very nicely. So again, very good resolution. And on the right here on the IR laser, we have the CD14 antigen, a very strongly modulated antigen. So I mentioned already earlier that the modulated antigen might be the most challenging to implement. And here we have a real isolated possibility, spectrally isolated possibility on the IR laser to have bright signals from the monocytes without any impact on others. But at the same time, the IR laser detectors are not subject to spillover from other laser detectors, so you can also very nicely detect here those dim expressors.
Great. It's such a good example. So let's then continue to talk more about the instrumentation, light management, and software, and let's focus a little bit more now on the software part of things. So we discussed measurement error being reduced, how the instrumentation can contribute to the sensitivity, and now we can talk about how the software might help to facilitate your workflow. I'd like to introduce a topic about the relationship between the voltage or gain and the fluorescence intensity. So on a photo multiplier tube, this is a nonlinear relationship, and so it's not easy to predict. And therefore, when adjustments are made to the voltages, it's required that experimental design and settings need to accommodate for that, right? It's something you have to determine empirically and iteratively. On a system that's equipped with avalanche photodiodes, there's the potential for the software to make use of that information, right? So we've got a really reproducible manufacturing process for the semiconductors. They're really well controlled, and therefore they're easy to predict, and as well as they're very linear. The response between the gain and the fluorescence intensity, or the response on that detector, is very linear. And so this is something that the software can be used to actually predict and facilitate workflow. So let's take a look at that next. If we are working in the SiteXpert software controlling a CytoFLEX instrument, we, during our experimental setup, can utilize the capability called gain-independent compensation. So what this means is that when you create your compensation, you can acquire the singles as much as you're used to doing and save the matrix or save the settings or the files to a compensation library. So then you go to your experiment, right? And you import the compensation into the experiment. A software window, a window will pop up in the software, and it'll give you some options. You can import the compensation matrix and actually recalculate it with the current gains that you maybe have been already working with, or you can just import the matrix and the gain as it was acquired. So these are new options that I don't think are customary for the flow cytometry workflow, but these are options that you have available when working with a CytoFLEX. And what we've been talking about up until now is the reason why. So the really nice thing is that you can adjust your gains on the sample if you need to, and the compensation will automatically adjust. So the next thing I want to show you is actually how that works within the software, right? So what we're going to see next is a short video clip within the CytoFLEX software where you'll see a compensation matrix, and you'll see a couple of plots. And the plots here, one of them, the user that's driving the software is going to access the gain tool from the menu ribbon, and then they're going to point out on the compensation matrix what you should be focusing on. They're going to actually run the sample and adjust the gain during that acquisition, and what you'll be able to see is the values in that compensation matrix adjusting in real time. So let's go ahead and play that video. So the cursor is now moving, pointing out the compensation matrix and showing you these values. And then once we grab the gain control tool from the menu, sliding it across the plot actually changes the acquisition setting or the gain setting, and the compensation matrix recalculated in real time. And that is basically something that you have affordable to you within the CytoFLEX software, which really is a very unique, powerful tool. And so that is a powerful tool within the software that I'd like to next demonstrate the utility of by reviewing with you the workflows for compensation. So that my intention to showing this video was to demonstrate the gain-independent compensation in action. And now let's look at how that translates into the workflow for generating a compensation matrix. So this is a traditional workflow. What we normally do is we prepare a series of tubes each tube is a single stained control that represents each color, each fluorochrome that you've got present in your panel. And you find a good setting for each one of those single stained tubes. Then you acquire each one of those at the settings that you determined. The compensation matrix is then calculated. The matrix is then imported into an experiment, and then you'll acquire your mixed stain sample using the settings that you had determined for that compensation matrix. You take a look at the data, you assess the data, then you decide in the workflow where that question mark is. This is where we're deciding, am I happy with the data or am I not happy with the data? If I don't think there's any adjustment needed, we're going to follow the path to analyzing the data, which we're ready to go. But if there is something that's not quite perfect, that we can't just do post-acquisition compensation adjustment for, it's something that actually really does need a gain adjustment for, we then need to go down the path where we have to rerun the mixed stain sample and make that voltage adjustment, then go back and reacquire every single one of those single colored stains at those new settings, recalculate the compensation matrix, and then look at my mixed stain sample again, assess the data, and come back to asking myself, am I happy with that or not? And if I'm not happy, I've got to go back through that loop and continue to follow the loop until I'm happy or until I'm just happy enough, and I move forward with my experiment. So it can be quite tedious and quite iterative, and the more colors you have in your panel, the longer this type of workflow can take. So on the CytoFLEX, it's changed everything, really. Well, it hasn't changed everything. You've got the beginning where it's the same, where you need to have a single stained control for each one of the floors that are represented in your panel. You can calculate the compensation matrix once you acquire those single colored stains at a reasonable voltage setting. And then you import those compensation settings into your experiment, and you can decide at that point whether or not you want to adopt the gains from your experiment or leave them the same as what you acquired during your compensation acquisition. But the thing here is that the software at this point could even just recalculate for you. You can then acquire your mixed stain sample, assess the data, and you're at that decision-making point now where you're either happy with the data or not. And if you're not quite happy with the data, you can put your mixed stain sample back on, and during the acquisition, adjust the gains. The compensation will recalculate automatically, and you're done. You don't have to go back and reacquire all of the single colored stain tubes, because The software can accommodate for the smart design of the instrumentation, and it can utilize that information to simplify the workflow and thus apply gain-independent compensation, which makes the workflow much simpler, especially the more complex situation that you get into with multicolor panel design. So the CytoFLEX platform really is flexible, and it also provides exquisite sensitivity, sensitivity for fluorescence, as well as small particle characterization and detection in a benchtop analyzer. There's an extensive set of repositionable bandpass filters that come with each instrument. You can upgrade within a series by activating additional detection channels, which would be like you can turn on a laser, you can add additional detector activation within a series. And it's powered by intuitive software. The CytExpert software is really powerful and is well flexible. That helps to, there's lots of tools in there that really help to make your multicolor panel design and analysis much simpler. And you can find more information at beckman.com/CytoFLEX. And we've also today talked about some resources, and we provided some resources in the webinar. So in the resources section, you'll be able to find the CytoFLEX Flurochrome chart that has the configuration diagrams for each model within each series. And then we've also provided the poster that demonstrates the 21 color, 20 marker immunophenotyping example that we showed data from today. And additionally, a poster from a couple of years ago at Cyto where a 27 marker was demonstrated on a 3 laser CytoFLEX, also demonstrating that if you're using good panel design, incorporating marker stacking and smart instrumentation, you can do deep immunophenotyping with a simple configuration as well. So I am happy now to take, see if Michael's got any further comments for us.
I just would like to summarize what we have been going along here. So bright dyes are not always the best solution. You really have to follow this Goldilocks principle, finding the optimal match of brightness for the antigen expression that you want to look at. And we have seen that the instrument characteristics with the spreading matrix are also very important to understand. So how sensitive can you be on a certain channel? How susceptible is a certain channel to spreading when receiving spillover from other dyes? The biology is really central. So, and this covers the expression profile, but also the relationship between those antigens that have these specific expression profiles. So I would like to remind you of organizing the panel in a tree-like shape to really understand best where you can allow to have spillover, where you should avoid it. How do these antigens relate to each other? I think that is pretty helpful. Yeah, and then of course I would like to point at the resource section like Maria that has done already. Here you find really examples that illustrate very well how you can follow these principles and how you can achieve powerful panel solutions here.
Right, and how the instrumentation can help to simplify the decisions you need to make, because sometimes you just have hard decisions to make.
Exactly. Yeah. So I think, Maria, we are at the point where we have finished our presentation. We would like to thank you for your kind attention and hope it was informative for you. Yeah, I think we have.
Anyone have any questions? We're here now to answer any questions that you might have.
Yes.
Thank you.
Please go ahead.
Thank you, Maria and Michael, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Maria and Michael will answer as many questions as time permits. Okay, let's get started. The first question is gain-independent compensation a new capability on the CytoFLEX?
Oh, good question. Actually, no. It's been capable since the very first release of the software and the hardware. So CytoFLEX has always been capable of being independent compensation. And if you would like more information on that, we can definitely help. So yeah, it's been there the whole time.
Great, thank you. Regarding an existing antibody panel in my lab, how could I check the panel performance and see if I am missing something due to suboptimal panel design?
I would like to take this, Michael. So this is a really, really good question. So once you have already something that you all have good experiences on, but maybe there is some hidden flaw in it that you might not have been aware of. So the best way to do that is really, from my point of view, look at the single color staining and see how each of the single colors influences the background on each other channels. And maybe you also do FMO controls. Look at these FMO controls not only from the angle that you inspect the channel where you have left out the dye, but also look at the other channels. What does leaving out a certain marker do to the background of the other channels? I think that is a very effective way to see if there is a combination of antigens that causes a loss of sensitivity on a given channel. So please use your FMO controls, looking at each and every channel and see how different is that as compared to the complete staining. That would help to see if you're missing something.
Thank you. What dyes do you recommend using for the IR laser?
Okay, I'll take that one. So there is the Biochrome 808 that was demonstrated in the poster, the data that we showed today, and that is a fixable viability dye, and that can be detected on the 8540 channel. And then believe that CD14 was labeled with Alexa Fluor 790, and that was detected on the 840-20 channel of the IR laser.
Great, thank you. Okay, next question. How can I measure and compare the performance of different antibodies against the same antigen?
So, Michael, I would like to take this one. I think there are two ways. So one way is you could just do single stains and see how well the separation is between the positive and the negative part of your profile. This is what typically is referred to as a signal-to-noise ratio. That's a good approach. However, if it comes to multicolor staining, I think it's better to think about what is called the staining index. So that is not like the signal-to-noise ratio, just building the ratio of positive and negative fluorescence intensity. Here you would look at the difference, so the delta of the intensity, and you would divide the delta by the broadness, the standard deviation of your negative peak. So you can well imagine, as you have a certain separation represented by a delta and fluorescence intensity, the effective separation will be impaired as the negative population will get broader and broader due to spreading effects related to the panel design. So this will really tell you within the application that you are aiming to build, how is the separation of the markers. And then you can also compare different positions of this marker. So you use different fluorochromes for this marker, so you have different spectral environments also for detection of this antigen. And this is how I would compare this when I really think about the design of panels. Think about the staining index.
Thank you. It looks like we have time for one more question. Could you further comment on why especially antigens with modulated expression densities can be difficult to implement into an antibody panel?
Yeah, I would like to take also this one. So modulated expression refers to a profile, an expression profile, where we would see cells that have a very high expression density, but also we would have cells with intermediate and also low expression densities. And this sets up a couple of requirements. On the one hand, you want to use a dye for that does not have a large impact on other channels, because we will have very bright events, and we want to avoid that these bright events cause spreading in the channel, which will make it difficult in these other channels to detect co-expressed antigens. But at the same time, we have very low expression densities. So the channel for the detection of the modulated antigen also should be clean of spillover from other channels. So we have basically both requirements at the same time, not to exert spillover, but also not to be receiving spillover from other antigens. That makes it a little bit difficult. There's not so much dyes and positions in a certain set of detector arrays that fulfill all these requirements.
Thank you. I would like to once again thank Maria and Michael for their presentations. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speakers via the contact information you provided at the time of registration. We would like to thank LabRoots and our sponsor, Beckman Coulter, for today's educational webcast. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.